This is interesting because once we broke the mold and realized that we could use gene therapy for things other than treating Mendelian diseases and cancer, we started with osteoarthritis, but most of my career I've spent in orthopedic department, so it was a natural to move into bone healing. And that's an interesting topic because you normally think of bone not needing any help to heal. If you break your arm, you'd expect it to heal unproblematically. And bone does regenerate unproblematically, except when it doesn't. And then it's very recalcitrant. Now, there's a product on the market that's a recombinant human bone morphogenetic protein 2. It formed the basis of something used in orthopedics called Infuse. And it works. It's an osteogenic agent, but the problem is it's hard to deliver. And in fact, it's a bit like the problem with osteoarthritis in a certain way. And it's difficult to get enough of it at the site of the lesion for long enough to do what it's supposed to do, namely heal the bone. So the way clinically it's used is just to put massive amounts of the recombinant protein into the bony defect, such that enough of it sticks around long enough to do what it's supposed to do, namely heal the bone. And the problem with that is it introduces side effects, and some of them are quite severe. It makes it prohibitively expensive, and also the clinical outcome isn't as good as advertised. It doesn't have the same potency as you might expect. So we started delivering it by gene delivery as a way of overcoming the delivery problem. So we're not fixing a genetic defect. We just want to get more of the stuff into the bone for long enough to do what it's supposed to do. And knowing that bones heal in a few weeks, we didn't think we'd have to use an adeno-associated virus as we are in the chronic condition of osteoarthritis. We just needed something that would express for a few weeks, and adenovirus seemed appropriate. What we found out is quite interesting because we went in thinking we needed to express as much gene product as possible for as long as possible to get the maximum healing effect. When we look closely, what we found is that you actually can heal bones quite nicely if you deliver a small amount for a limited period of time. And the key thing seems to be not that you have a lot of protein there, but the protein is synthesized by a cell in the location. And there's something special about the way a local BMP-2 producing cell communicates with its environment that's different from what you see when you add a big swathe of recombinant protein. And the thing about that is that having realized and appreciated that you actually don't need very much, but it just has to be done in the right way on a cell basis locally, we've actually gone over to using chemically modified messenger RNA in conjunction with colleagues in the Netherlands and in near Munich in Germany. And what we found using this chemically modified mRNA that encodes the same protein, BMP-2, but can now take advantage of the fact that it can be delivered by transfection, and we don't need huge gobs of gene expression. We're getting very dramatic bone healing using this same technology they're using for vaccinating against COVID. So we're quite optimistic about this, that it could go the distance.